it's been fantastic. So thank you for that, for making that happen. And I think it goes really beyond access. It is probably really one where it starts to shape how we do research in general, and that's phenomenal. So I'm here really speaking on behalf of about 70, 80 people in theme 1. We are tasked with looking at cancer biology, which is a massively wide field. So we approached this in 7 working groups. And really our overall objectives were that we wanted to push the boundaries on some of the tough questions that remain, and there are many, in pediatric cancers, doing and supporting the fundamental research that's necessary, and then drive this all the way to translation and to the interface with clinical care, with patients, and to make sure that this component is squarely embedded in ACCESS. And so we divided it into 7 working groups. As I said, we have 3 working groups that focused on disease areas and fundamental problems there. There we identified brain malignancies, leukemias, and sarcomas as the ones we want to focus on first, because we can't do it all at the same time. And then we identified really one key component that we need to work on, and that's data sharing, biospecimen collection and sharing, and access to this. And we are not the only one who identified that as a big need, but really that came up across all the themes, and that's why we'll be talking about this tomorrow. And I'll speak a bit about what we've been doing there tomorrow. Then we identified the question of diving into the pediatric cancers in a bridge between research and supporting patient care immediately in model systems and trying to understand them in a personalized way. And that's where we already started and got funding for the first project. It's called the Pediatric Cancer Modeling and Mechanisms Group, where we really bring together patients with their individual tumors and open questions, and scientists, researchers across the country, possibly even international, with knowledge in those areas, and to try to answer those very personal open questions and in trying to find solutions there. We also realized that we have been quite successful over the last couple of years with getting sequencing and personalized treatment more or closer to becoming routine care, but that it's not always successful, and it leaves many open questions. And that we need a sustainable pathway to identify the next assay technologies and figure out what's coming next. And there we identified liquid biopsies and circulating tumor DNAs and proteome analysis as the next level we want to work on. A proposal for this is being evaluated. And of course, we also wanted to make sure that there is a pathway for the one that comes after this, so that we can do better in this and really serve the patients well in Canada and beyond. And I think I had them all. And really, I think what is the key to having everybody here and people online now is that we can start to talk about how can we work together. And there are many areas, as you see in this complex figure down there, where we intersect with the other matrices, but so far we had not the opportunity and time to really talk to each other. But we very much want to do so, and are very much looking forward to working together and making this an integral